Thank you for introduction, and also want to thank the GWICC and AHS for the organization and invitation. So today I want to share a molecule we have been working in the lab for many years. We thought it may be a potential therapeutic target for ischemic heart disease. So we all know the ischemia precondition is especially the second window of the precondition is a very is a very potent mechanism of cardiac protection. However, it is very challenging to induce the preconditioning in vivo without tracking the ischemia. So it's very impractical in the clinical. However, we thought if it could activate the relevant survival signal pathway, it might be a very appealing therapeutic for preventing or therapy of the ischemic heart disease. So during our previous study, we found VCP, which is ATP as a social protein, increased in the second window preconditioning model about 3 to 4-fold increase. Also, we found mutation or inactivity of this VCP result in dilated cardiomyopathy and also heart failure. In vitro study, we overexpressing this protein protect against the apoptosis of cardiomyocytes. So we are interested in this molecule. So we first generated cardiac-specific knockout mice of the VCP. For all the homogenized knockout mice cannot survive. However, the heterozygous knockout mice survive with about 40% of the reduction of the VCP. And these mice, the homozygous, heterozygous knockout mice, progressively developed the cardiac dysfunction and increased the mortality. Almost all of the mice, even the heterozygous knockout mice, have been died around the 10 to 12 months. This data indicated that VCP in the heart, in the cardiomyocyte, is very important for the cardiac function and the development. So next we want to know the increased cardiomyocyte VCP in the second window of the preconditioning have the protective effect, or just the secondary increase during the procedure. So we generate a transgene mouse. We have the different founds with the different level of the overexpression. And we pick up the one level really similar to the increased level we found in the preconditioning model. So about 3 to 4-fold increase. And we induced the IR in ischemia reperfusion injury in these mice. We found the transgene mice with a 3 to 4-fold increase of the VCP expression significantly protect the heart against ischemia reperfusion injury, which is very similar, is comparable to the preconditioning. And we also found in vitro, also in vivo, increased VCP induced the INOS, which is the well-known the end effect of the second window of the preconditioning. And also, we found this increased INOS by the VCP can be abolished by the inhibitor of the NF-kappa B. And also the cardiac protection mediated by the VCPs also can be abolished by the INOS inhibitor and also the NF-kappa B inhibitor. Another side, the sinus VCP also reduced the INOS expression and impaired the cardiac cell survivability. So that is background summary. Is the increased VCP involved in the protective mechanism targeted by the preconditioning, and deficient VCP is associated with the cardiomyopathy and the cardiac dysfunction. Cardiac-specific overexpression VCP in transgene mouse imitate the cellular mechanism induced by the second window of preconditioning and reproduce the cardiac protective effect of the second window of preconditioning, offering a emptive conditioning to the heart and highlighting it as a promising therapeutic target. So then we'll continue to figure out what the mechanism. First, we found the VCP-mediated activation of alpha-kappa B and Aionos required activation of the AKT. First, if we use the inhibitor of AKT, they can abolish the VCP-mediated increase of the alpha-kappa B and Aionos. And then we found in the transgene mouse, overexpression VCP can significantly activate phosphorylation of AKT at the 473. And in the other side, if they in the isolated cardiomyocytes also induced the dose response increase in the phosphorylation of the AKT 473. And further, we used the knockout mice. We found the, I don't know why it disappeared. If knockout mice, the phosphor AKT is at 473 is totally disappeared. And also we found this mechanism may link to the mTOR complex 2, and also they have increased the PP1. This is the protein specifically dephosphorylation of the AKT, and also increased the P47, which is the cofactor of the VCP. So this is a summary of the VCP activity may related to the P48, PP1, and then eventually through the phosphor AKT. So next we try to see what the mechanism, if the VCP mediated the cardiac protection rely on the Aionos expression. So we crossed the VCP knockout transgene mouse with the Aionos knockout mice. We get the biogenic mice. So first we found increased VCP inhibit MPDP opening in the heart. And this effect were abolished by if you donate the Aionos from this transgene mouse. Further mechanism study, we found VCP inhibit the MMP opening through the protect the calcium uptake into the mitochondria, then reduced the calcium node in the under the stress condition, and also the preserve the ATP production. That means through the reduced consumption of the ATP during the stress. And the mechanism also the molecular mechanism related to the VCP can increase mitochondrial CQ1 degradation, where the proteasome they increased the proteasome activity and also the items, the components, and also they have through the specific binding with the Perkin molecule. So in summary, VCP inhibit MPDP opening and repress ATP consumption through the monitoring calcium overload in the stressed condition. On the other side, the VCP also regulated the mitochondrial respiration in the heart via the Aionos. We found the mitochondrial respiration, especially the state 3, increased in the transgene mouse, which would be abolished by the Aionos knockout. We also found the VCP increased the mitochondrial translocation and also increased the Aionos mitochondrial distribution in the heart. Then we further investigate what the mechanism, the VCP Aionos in the mitochondria, and we did the Aionos. We did the cardiac protein astronization. We used the mass spectrum, and we found the majority increased the SNO is mitochondrial protein involved in the mitochondrial respiration, metabolism, and the calcium regulation and ear stress, and majority related to the mitochondrial and metabolism functions. And we further used the different strategy to detect overexpression VCP, activate VCP itself, and GABA-DH SNO in the heart, which is not rely on the inos. We also used the VCP transgene mouse and the biogenic mice. We found overexpression VCP can increase the SNO of the mitochondrial protein, especially the complex 1 and 2. However, this increase was abolished by the inos knockout. We also further identified the domain of the N-terminal of the VCP is the major domain of the VCP SNO and also the SNO function of another protein. I will not probably introduce too much about this part. Also, we generated, based on this, we generated VCP DIM transgene mouse, which is a mutant. It deleted the N-terminal to see if they have the effect of the cardiac function. In summary, the VCP mediated the SNO depends on the N-terminal and through the regulation of the cyto and the nuclear translocation. This is, since it's already published, I will not give too much detail about this one. So in summary, the VCP have, we also found have the, a lot of the other function in the ER and the mitochondrial associated degradation. So here is just the model we found probably VCP will target both ischemia and reperfusion, induced the cellular mechanism, then increased the cell survival and prevent or attenuate infarction and also the poor remodeling. So I hope I can have some message to home. First, the VCP is a key mediator of the second window of the preconditioning. Increased VCP reproduce the cardioprotection effect of the second window of the preconditioning. VCP cardioprotection associated with the inos expression through the recognition of the mTOR complex 2, PAKT473, pro-survival signaling. And the VCP inos repression IR injury by enhanced ATP production and inhibit the MPDP, sorry, this is type opening, via the modulate the cadmium overload and also through the SNO of the cardiac protein. Cardiac protection confirmed by VCP rely on the 10-terminal domain. So all of this work is really rely on my previous and current lab member, and also my mentor and the collaborator, and also the funding from NIH. Yeah, so I just moved a new institution. We are still recruiting postdoc, and if the graduate student, if you are interested, welcome to join us at the Phoenix. Thank you very much.